Biology Part 99. In order to maintain the impulse conducting functions described above, the neurons require a number of specialized internal structures. Such structures contribute to the metabolism of the neuron, allowing the single nucleus in the cell body to exert metabolic control throughout the dendrites and the long axon. The special structures present include nasal granules in the cell body and neurofibrils, which also occur throughout the cytoplasm. Each nasal granule consists of membrane of rough endoplasmic reticulum, which enables the neuron to be particularly active in protein synthesis, manufacturing within the cell the transport of these proteins and other substances produced in the cell body. It seems to be carried out by neurofibrils and both proteins from the nasal granules and glycolipids from the Golgi bodies. They are packed into the membrane-bound vessels before transport takes place. And while the neurons are the most important functional components of the nervous systems, they are not only all cells present, but in addition to the blood vessels, connective tissues, there is always a mass of glial cells, collectively known as the neuralgia, while some of these glial cells, such as the Schwann cells, are known to have special functions. The majority of it seems to be packing cells, giving mechanical and possibly metabolic support to the delicate network of neurons carrying the nerve impulses. Electrical properties of neurons. So far, the nerve impulse has been rather vaguely described as an electrical message. The first step towards the deeper understanding is to consider the electrical properties of neurons. These electrical properties have been studied using tiny probes called microelectrodes. The commonest type of the microelectrodes is the minute glass pipette filled with the concluding electrical signals detected by microelectrodes. So they are usually amplified and then passed to an oscilloscope, which displays the signals in a visible form. The tip diameter is a class microelectrode, which is often less than 1 and is appropriate technique. It can be placed inside the cell body or axon of any suitably sized neuron. The cell body of the vertebrate motor neuron is about 50 to 70 in diameter and therefore large enough to be penetrated successfully. However, much larger neurons are found in some invertebrate animals, particularly molluscs. The squid, for example, has a number of joint neurons with awards 1000 1 mm in the diameter and several cm in length. These joint axons have contributed significantly to the present underlying of the nerve impulse. Well, the figure shows the simple experiment which can be carried out using the squid joint. The squid joint axon. The axon would normally be dissected out and placed in a bath of fluid for the purpose. The microelectrode mounted on a micromanipulator for the fine adjustment is maneuvered until it penetrates the axon. When this happens, there is an immediate change in the oscilloscope We'll talk about this in the later video.